Now dear students, today we will go through the second phase of the post-fertilization events. It is the ovules maturing into the seeds. Seed or fertilizer ovule is called a seed. Morphologically, ripened ovule is seed. Seed is a matured, integumented megasporangium. All the structures which are present inside the seed coat are called or termed as a kernel. Typically, matured seed is having three main parts: the seed coat, then the endosperm, and the cotyledon. So mainly it has three parts. A seed contains mainly three parts. The outer one is the seed coat, then next to it is the endosperm, and here it is the cotyledons. Now the structure of the seed. Seed coat formed from integuments of ovule. Its outer layer is testa here. Its outer layer is testa, where this inner one is the tegumen. The micropyle remains as a small pore in the seed coat which facilitates entry of the oxygen and water into the seed during germination. It protects the embryo. Endosperm. It may be present or absent. Then the third one is the embryo, which gives rise to the mature plant and maintains continuity of the this generation. Now it is the structure of some seeds. Here it is the bean seed. These are the two cotyledons. Here it is the castor. It is seed coat. It is endosperm. These are the cotyledons. This is the hypocotyle root. This root axis. This is the shoot apical meristem here. This is the onion, and this one is the maize seed. Now there are three types of seeds. Endospermic, they are also known as albuminous. Non-endospermic, also known as non-albuminous, or as well as the X-albuminous. And then the last one is the perispermic. Now the endospermic. Here the food is stored in endosperm. The endosperm tissue is utilized during the germination of seeds. When a baby plant is there present, it uses this endosperm as the nutritive tissue. The cotyledons are thin and membranous. Examples are the wheat, rice, maize. These are the monocots, whereas the cotton, the sunflower, these are the dicots. Now the non-endospermic. Such type of seeds do not have an endosperm at maturity. Endospermic tissues are absorbed during the development of the embryo. The absorbed food materials from the endosperm is stored in cotyledons. That is why they become so large and fleshy. The examples are the gram, pea, groundnut, castor, as well as the sunflower. Now the perispermic seeds. Seeds in which remains of nucleus is seen. The residual persistent nucleus is called perisperm. Example, black pepper or the beet. Now the seed dormancy. Dormancy is a state of inactivity when the seed is not able to germinate. The time or the period between the maturation and the germination of seed is known as dormancy period. Now the importance of this dormancy. Dormancy helps embryo to pass on adversarial environmental conditions, suspends all kinds of growth and development, and also keeping embryo inactive during the unfavorable conditions till the favorable conditions reach or the arrival. Now the reasons of seed dormancy. First of all, the impermeability of seed coat. In many species of families like the Leguminosae and the Convolvulaceae, their seed coats are impermeable. That means it does not allow moisture and oxygen to go in. Here the seed coats are thick and hard, and cell wall contains layer of the lignin, which makes it impermeable. Such seeds take more time for germination. Then the germination is caused by the action of the soil microbials, or we can say the soil bacteria. Next is the dormant embryo. In several species, the embryo is not completely mature. When seed ripe, even it fails to germinate in the favorable environmental conditions, or even seed coat is removed. Such embryo is referred as dormant embryo. Here, again the dormancy is broken down by the enzymes and some chemical reactions. Third one is the germination inhibitors. Seed germination of some seeds is sometimes checked or prevented by the presence of some chemical compounds called germination inhibitors, such as ferulic acid. It is present in tomato juice, coumarin, abscisic acid, and para-ascorbic acid. This condition is generally found in xerophytes. These germination inhibitors washed away with water. This is called leaching. So as a result of this leaching process, these seeds become now able to germinate. Now the methods of breaking up seed dormancy. First of all, scarification. In this method, hard seed coat is broken so that water and oxygen can enter into the seed coat. It can be done by first A, making minute hole at the pores with the help of pointy sharp apparatus on the seed coat. Secondly, seeds are rubbed on hard object so that seed coat becomes thin. Next, partial degeneration of seed coat. It is done by the sulfuric acid. Next, stratification. Here the seeds are kept at low temperature in the presence of oxygen and water for some time so the embryo can complete its maturation. Very important is the light treatment. Seeds which are affected by the light are known as photoblastic seeds. Seeds germinate in presence of light are positively photoblastic seeds, whereas the seeds germinate in absence of light are termed as negative photoblastic seeds. Another type of seeds, those seeds which are not affected by the light, they are known as non-photoblastic seeds. Continuously, the dormancy of photoblastic seeds can be broken by the treatment of red light. The phytochrome red, it absorbs the red light and converts into phytochrome 4 red, which increase the germination of seed. Here you can see the several combinations which affects on the germination of seeds. When the seed it is treated with the red light, it germinates. When the seed is treated first the red light, then the 4 red, here no germination takes place. But when the seed is treated with this red light, then 4 red, that again a red light exposure is given, it germinates. But those seeds, where first the red light, then the 4 red light, then again these red lights, they are then followed by the 4 red lights. Here no germination takes place. Now the next one is the alternative temperatures. Seeds are treated with high and low temperatures, alternatively at high pressure to increase the germination of the seeds. For example, seeds of melilotes, that is sweet clover, and alfalfa treated at 2000 atmospheric pressure at 18 degrees Celsius. Growth hormones. Seed dormancy can be broken by treatment of gibberellic acid. Viability. Viability is the existence of life in a seed. It can be tested out by 2,3,5 triphenyl chloride. The embryonal axis of living seed becomes pink in color in the solution of TTC. Seeds of a large number of species live for several years, whereas some remain alive for hundreds of years. The number of seeds in a fruit is generally equal to or less than the number of ovules in an ovary. It never exceeds the number of ovules. Orchid fruits contain thousands of tiny seeds, whereas the parasitic species like Dormancy and Strega also contain many seeds. Now the advantages of seed to angiosperms. Seeds have better adaptive strategies for dispersal to new habitat. It has sufficient food reserves for nourishment of young seedlings. Protection is provided to young embryo by the hard seed coat. Generate new genetic recombination as it is a product of sexual reproduction. Seeds, the base of our agriculture. Seeds form the base of our agriculture as they shows dehydration and dormancy. Now the seed germination. Germination is the ability of a seed to produce a seedling in presence of favorable environmental conditions. Those favorable conditions are the adequate moisture, oxygen and stable temperature. Now the types, depending upon the behavior of cotyledons, germination is of two types. But viviparous germination is also found in some cases. So the types are the epigeal, hypogeal and the vivipary. Now here, in epigeal germination, due to hypocotyledonal growth, cotyledons are pushed out of the soil. Here, these cotyledons, they are pushed out of the soil. This type of germination occurs in castor, tamarind, bean. In some cases, these above ground cotyledons become green leafy-like. Cotyledonary leaves and perform photosynthetic functions till the seedling assumes independency. This is the epigeal germination, how it takes place in case of the castor. Now the hypogeal germination. In hypogeal germination, due to the growth in epicotyle pilumule, this pilumule comes out of the ground and cotyledons remain underground. This type of germination occurs in most of the monocotyledons and a few dicotyledons. Commonly, maize, rice, wheat, coconut, gram and mango. Now the vivipary. Viviparous germination or the vivipary is a special type of seed germination which is characteristic of mangrove vegetation found in muddy, saline conditions. Here is no resting period of embryo and germination occurs inside the fruit, while it is attached to the parent plant, that is in situ germination within the fruit. Examples are the rhizophora. Here it is a rhizophora. It is its example. Now, factors affecting seed germination. Firstly, it is the moisture or the water. Seeds absorb water and swell up to increase in size before the germination. Water is absorbed through seed coat and micropyle. The following effects have been observed in seed due to absorption of the water. The seed coat becomes soft and absorbed water reaches to the cells of embryo. When the ratio of water increases in the cells, the protoplasmic contents become more active. Secondly, the increase in the size of embryo results in the breakdown of seed coat. Thus, radicle and pilumule easily comes out. Next, the softness of seed coat increases its permeability and are and also increase rate of exchange of oxygen and carbon dioxide. Another one, the rate of respiration also increases in the active cells of embryo. It causes the activation of enzymes which change the stored food into the dissolved form. Secondly, oxygen. Oxygen is essential for oxidation. It releases the energy from organic substances, thus leads to cell division and elongation besides many other biological processes. Now the temperature. The suitable temperature is important for seed germination. The favorable temperature is 20 to 25 degrees Celsius for the germination. Now the food or the nutrition. The embryo depends upon stored food materials in cotyledons or endosperm in the germination period, up to the formation of primary root from the radicle and first leaf from the pilumule. Light produces different effect in different variety of seeds. Most of the plants do not require light up to the formation of first leaf. Rate of germination is very fast in absence of light. For some plants, light is very essential for germination. They will not germinate in the dark, like the lettuce and the mystery toe. Now the ovary maturing into fruit. This is the last post-fertilization event which takes place here. Fruit, it is a ripened ovary. The wall of ovary forms the wall of the fruit, which is called pericarp. Now types. Fruits may be true fruits, fruits which develop from the ovary, like the mango. False fruits, fruits which develop from other floral part, like thalamus, along with ovary, like the apple, strawberry. Now the parthenocarpic fruits, fruits developing without the process of fertilization. These fruits are seedless and can be produced through application of growth hormones like auxins, and example is the banana. Now this was all about the post-fertilization events. In the coming up, we will go through the genetics. First of all, we will study the Mendel, his work, experiments, and Mendelian laws. Thanks.